And this is important when you conduct studies, because you then need to know that not only those that are sick have immune response to the virus, but also those in the environment, those who are also cared for them, have immune response. So that when you vaccinate, you then need to know what level of vaccination you need to be.
This is Immuno Africa, a podcast dedicated to spotlighting African immunology research. Here, we amplify the stories of Africans researching the immune system to tackle the continent's growing burden of infectious and non-communicable diseases. I am Ralph Okete, a biochemist and science communicator in Nigeria keenly interested in research on immune system. On Immuno Africa, I explore and communicate research through the eyes and lives of Africans who do them. Whether you are a seasoned immunologist, a burgeoning researcher, or even an untrained person, I hope the time you spent here rewards you remarkably. Enjoy.
In this episode, I'm joined by a Nigerian scientist who has trained in some of the best universities in his home country and the United Kingdom. Dr. Chinedu Alphonsus Ugwu holds a Doctor of Veterinary Medicine with distinctions from the University of Ibadan, Nigeria, a Master of Science in Integrated Immunology from the University of Oxford, UK, and a Doctor of Philosophy in Veterinary Sciences from the University of Cambridge, UK. He currently lectures in the Biological Sciences Department at Redeemers University, taking undergraduate and postgraduate classes in immunology, virology, bioethics, biosafety, and biological techniques. He also doubles as a postdoctoral research fellow at the African Center of Excellence for Genomics of Infectious Diseases, ACEGID. At ACEGID, he leads several projects, one of which is a Biotechnology and Biological Science Research Council BBSRC funded project on One Health and accelerating vaccines for Ebola and Lassa Fever. His research focuses on understanding how the immune system modulates the outcome of viral infection by elucidating the correlates of protective immune response to viral infections in West Africa. He aims to contribute to vaccine and therapeutic designs, diagnostics production, and inform public health policies for the diseases.
My name is Chinedu Alphonsus, and I'm a senior lecturer at the Biological Science Department with Redeemers University, and I'm also a postdoctoral fellow at African Center of Excellence for Genomics of Infectious Disease, ACEGID, here at Redeemers University. In ACEGID, I coordinate most of the immunological studies. We look at how the body responds to viral diseases such as Lassa fever and coronaviruses that cause the COVID-19 disease. And I teach both undergraduate and postgraduate courses in biological science such as immunology, bioethics, biological techniques. I think that's me in a nutshell.
Yeah, thanks for that introduction. So I think something that first caught my attention in your profile where I noticed you attended some of the top schools in different regions, viz. Oxford University and Cambridge University. So I think I should just ask about, what was your experience schooling in some of these top schools? What was it like, being in these schools to take your studies and conduct research?
So my undergraduate was in veterinary medicine at University of Ibadan. And then I got a scholarship to do a master's in immunology at University of Oxford in the UK. And then I also proceeded to do my PhD in viral immunology at the University of Cambridge, the Department of Veterinary Sciences. About my experience, it's better when you experience it, but one thing I would say is, if I have to do it again, I will always go to those places and do it again. It's intellectually tasking, but you have the best support system in those three places. I don't know what UI is like now, but I had a good education in Ibadan, and it prepared me very well for my education abroad. And in both Oxford and Cambridge, I think for me the strongest thing take home is intellectually stimulating and tasking. However, you have the best support system. There is every human being, everything you need to support you to succeed. And I think that's my biggest take going to those places. Thank you.
Yeah, thanks. I think the big question now that I would want to ask is, why you delved into immunology research. Your first degree was in veterinary medicine, and then for your MSc, you did an MSc in Integrated Immunology at the University of Oxford. So what got you interested in immunology research and not any other kind of research?
I don't know why I'm laughing, but I'm good. I don't think I've ever said this part of my story, but now
Okay.
you've asked me, I'll say it.
Yeah,
So
share.
it's interesting. I did veterinary medicine, and one of my mentors, and still my mentor, is Professor Victor Anosa. He was the one that taught us veterinary immunology, I think in my fourth year. He's an inspirational lecturer. He doesn't come to class with notes, and he speaks science. And I remember sitting in that class and listening to him describe the immune system and how it's such an important system that protects us from disease. And then the interaction of that system with pathogen. So the system is so well organized that there is a path that should fight this type of pathogen. There's a path of fight. It's like you do 1 plus 1, it becomes 2. 2
Mm-hmm.
plus 2 has to become 4. It cannot be any other thing, it has to just be 4. And then when there is a challenge, the system has a backup to regulate itself. I just felt fascinated by how he described it. And I said, finally, this is my niche. And then the second part of the story is I remember doing veterinary surgery. I think it was in either 600 ago or 500. It's a long, stressful, but enjoyable process of having to operate on the animals. And I also remember one of my teacher looking at me and telling me, "Why is your eyes red? You look permanently sedated." I
Yeah,
laughed. I felt like I was tired and stressed and bored.
yeah.
And I just realized that yeah, I'm a veterinary, I read veterinary medicine, but I don't think I will be the best surgeon because I felt stressed. But I felt differently whenever Professor Anosa comes to class to discuss immunology. And that told me this is my niche. I enjoy immunology. I'm fascinated by that interaction. And I told myself, you know what? If you want to succeed in science, you got to follow what's your interest and what
Mm-hmm.
fascinates you. And that was it for me. I told myself, let me focus on this immunology. And so far, I don't think I've regretted the jump. It's been an exciting moment, and you can't get bored with immunology. Don't worry about the big jargons. It's an interest. There's always something fascinating. There's a new disease. The immune system has to do something, and that cemented my interest in immunology, and it is what it is till now.
Yeah, yeah, really interesting story. I think there's this special allure to the field. You know, it's attractive, it's fascinating. There's, like you said, there's always something new to learn, and it's complicated but also organized system. Thank you for sharing your story. So you've been working in immunology ever since, from your MSc to your PhD. I know you've worked on Lassa fever, like the virus, the Lassa virus. You've worked on Ebola virus. You've worked on also you worked on SARS-CoV-2. That's the popular coronavirus. And I know you've had some fascinating findings from your study. So just give us a general overview of what your research has been on.
So I'm gonna for my like I said, my background is immunology. For my
Hmm.
PhD, I studied there's a virus that is called lymphocytic meningitis virus (LCMV). It's in the same family with Lassa fever, and Lassa fever is an important viral hemorrhagic fever that is endemic in West Africa, most especially Nigeria. So LCMV is not as dangerous as Lassa fever. It causes problem in those that have weak immune system, but in normal healthy people, it doesn't cause problem. So I use LCMV to understand the immune interaction with these viruses, and that's what then most of the knowledge and the skills I gained from LCMV is what I have now translated into working on Lassa, which is an important virus disease in Nigeria that causes viral hemorrhagic fever. So my work on Lassa fever is mainly understanding the immune interaction with this virus. There are different strains of Lassa fever. You have lineage 1, lineage 2, lineage 3, lineage 4, and the other new ones people are discovering now. The interesting thing of this lineage is distributed by geographical location. So, for example, lineage 1 was discovered in Nigeria, and the Lassa Fever, I think, is in Maiduguri. And then lineage 2 is commonly seen in the southern Nigeria. And somehow in our lab here at ACEGID, I think sometimes in 2018, we found out that the lineage 2, there are two types of that lineage 2 again. There's the one in the southwest, we call the lineage 2A, and then there's one in the southeast, which is lineage 2B. And what we think is that because of the river, I think it's the river Niger that separates these two places. The reservoir host, which is the mastodon, the giant rat, because it couldn't move around. So this is why the strain lineage 2A stayed back in the southwest, while lineage 2B remained in the southeast. So, and then lineage 3 is also found mostly in the northern part of Nigeria and then middle, I call it middle belt. And so those where you find the common. So the interesting thing about all of this is this disease, sometimes, I'm going to backtrack. This disease was neglected prior before 2018. There was no interest on Lassa fever. So people get sick. Sometimes you think, because it gives out similar symptoms like malaria. So you think you have malaria and then you treat malaria. Some lucky ones get better because if you're managing the symptoms, while the unlucky ones will die. And sometimes you think it was malaria that killed the person or typhoid. Meanwhile, it was Lassa fever. So it was not until the big outbreak in 2018 that the WHO now interest in the disease and research, and there's enormous effort that has been made. So most of our own work is looking at understanding the type of immune response that people produce when they are infected with the Lassa fever, and also understanding the diversity of the viruses around, and then most especially what happens in the reservoir host versus what happens in the actual host human.
Thank you so much for that breakdown of your work. I think we should just delve quickly into one of your recent publications where you tried to study the virus in people who had recovered from the disease and people who had been exposed to the disease, and to understand how the immune systems were interacting with this virus, and to look at how you could explore that understanding to tackle the disease. So I would just like you to tell us about that recent study and your findings.
Okay, thank you. So what we did is in 2018, which is when we started this project, and I'm going to be grateful to the funder. This was funded by the BBSRC in UK. So they provided us funds to look at one, the immune correlate of protection. What are the features of protective immunity from Lassa fever? And then secondly, the diversity of the virus. So most of my work was focused on those that survive Lassa fever. What does their immune response look like? So for immunity, we have what we call antibody, and then we also have what is called T cells. So these are what helps us to fight the infection, like viruses, like Lassa fever. So the aim was to look at what kind of antibody do people make, and then what kind of T cells do people make. And we look at this in those that have survived the disease, and then those who cared for them. That's what we did. So for these studies, we recruited in the two major sites that Lassa fever is very endemic, in Abakaliki and OVEL. The third site that we should have recruited was Irua, but unfortunately there were security challenges, so we couldn't get survivors from Irua. So we limited the study to Abakaliki and OVEL in Ondo State. So what we did, we will set up a clinic for Lassa fever survivors. This is the thing that was also very important in these studies that we didn't mention in the paper we published, is that we have these two sites to set up what we call the survival clinic. So those that survive Lassa fever, there's a lot of stigma to some of them because the disease can kill you and it's also contagious. People tend to stay away from these people. So what we set up that survival clinic is so that they will come to clinic and they will know any challenges they are facing post-survival. And that is something we are working on. Hopefully, we will come out with that data very soon. So when we recruited these survivors, and then also we also recruited those who cared for them. So we also want to understand why is it that those that had this Lassa fever and then those who cared for them didn't get the disease. So some of the intrinsic findings we got from the paper is one, that those who survived Lassa fever and those who cared for them, which we call the contacts or the exposed contacts. Some of these exposed contacts include the clinicians who manage these patients. They include the family members who also took care of them when they go home or when they were at home, and then other members around when they had the disease. The interesting thing about this contact is that they never had any positive Lassa diagnosis, both by PCR, which is what we normally use to diagnose them. So, but when we then measure their T-cell response and then antibody response, interestingly, we have the same magnitude of immune response between both the survivor of the disease and the contact. They made comparable amount of antibody. They also made comparable amount of T cells. So what that tells you is, what we think that explains that is that why those that survived the disease were infected, they must have been infected with maybe a level of the virus that's able to make them to fall sick, which is why they went to the clinic. However, the contacts, they must have been exposed. They may have had the infection, but they may have had no dose of infection, which their immune system were able to manage, and it didn't end up becoming disease. And which is why when they get to the clinic and tested for the virus, you wouldn't see. So that was one of the first interesting things. And this is important when you conduct studies, because you then need to know that not only those that are sick have immune response to the virus, but also those in the environment, those who have also cared for them, have immune response. So that when you vaccinate, you then need to know what level of vaccination you need to give. The second finding we then found, which was also very interesting now, is for the antibody response. These antibodies are what, like I said, what the body uses to fight disease. So what we saw is that it's one thing to make these antibodies, but it is also, these antibodies will recognize the virus, but it's also another level that those antibodies is able to fight the virus, so neutralize the virus. So we call it in immunology. You make a binding antibody, which is an antibody that can recognize the pathogen. And then the second layer is if those your antibody that you are making, which can recognize the pathogen, is able to neutralize the pathogen. So it's possible for you to have a binding antibody, but will not have a neutralizing antibody response. So, and why that is important is, if you have a binding antibody response, but you don't have a neutralizing antibody response, then it means that even though you have an antibody response, but your antibody is not neutralizing the virus. One good advantage for such is that it can help in diagnostics. So you can use it to separate those who have an active infection and those who just have a passive infection, non-symptomatic infection. So what we found out is that while the survivors made neutralizing antibody response, and again, this neutralizing antibody response was not, remember I mentioned initially that Lassa virus have different lineages, lineage 1, 2, 3, and 4, and other ones that has been discovered.
Yeah.
But for our work, we limited our assay to only lineage 1 to 4. And what we found out is that those survivors that made neutralizing antibodies, the antibodies were able to neutralize the pseudotype we use for all the 4 lineages. And then you had the strongest neutralizing antibody response to lineage 2, which is the commonest strain you have in the southern Nigeria, because most of our work was conducted in southern Nigeria. So that makes sense. But then for the contacts, who are, again, never tested positive for the virus, but they have binding antibody response. They didn't make neutralizing antibody response. So all that we saw for the contact is that they had binding antibody response. And the take home we'll now think is that it's most likely that having a disease makes your antibody neutralize, helps your antibody to helps you to develop neutralizing antibody response, compared to someone who have non-symptomatic infection, because the immune system was able to clear the virus before it becomes a disease. So that's important to know. Even though the contact has antibody, most of the antibodies were not neutralizing compared to the survivors who make neutralizing antibody response. Another important finding again from that paper now, especially like I said, there's the antibody and there's the T cells. What we looked at is we found out that for the T cell assay, we use what is called ELISpot. And interestingly, we remain the only place that you could do this ELISpot to measure T cell response in Africa. And I don't know anybody doing it. So this is one of the things, one of the things we developed during this project. We developed an ELISpot assay for Lassa fever. So what we do is we computationally generate the peptides of the virus. For example, the GP protein of the Lassa virus and the nucleoprotein. We generate it in the lab and we will synthesize it. So what would then the assay involves you collecting cells from a known survivor and then giving it to this peptide. The immune system is such an interesting system that it keeps memories when he has been exposed to a pathogen. So because this patient survived Lassa fever, they must have had T cells that remember that they have suffered from Lassa fever before. So when you give them this peptide, those T cells will become activated quickly and they will start producing what we call cytokines. This cytokines is interferon gamma. And those are what the T cells use to fight the viruses. So you can measure the amount of interferon gamma produced. So if you have not seen a Lassa fever before, then your T cell, when they are given the Lassa peptide, they will not make interferon gamma. But if you've seen it before, you will make it. So what we saw from our research is that both the contact and also the survivor, they were able to produce interferon gamma when they were given this Lassa peptide. And that is interesting, meaning they have been exposed to the virus. But the second layer to it is that we then looked at which part of this virus induced the strongest T cell response. And we can find a part on the GP protein of the virus and the part on the nucleoprotein of the virus. Why is this important? This is important because when you now design vaccine, because for those that want to design vaccine, what you do in designing vaccine, you take part of the viral protein and then design it in such a way, and that's what you then use to vaccinate the person so that you can make immune response to it. So what our study can tell you now is that if you want to design a vaccine targeting the GP protein of the virus or the nucleoprotein, we can tell you the exact part that you design your vaccine that we are sure will induce a very good immune response. That's another interesting finding we saw. And I think the last part is that we also looked at our cohort of these survivors over time. And what we see that the early survivors, those that have, I think it's over a decade, that they don't usually have any immune response, even though they are survivors of Lassa. But if you survive after a decade, you don't make both antibody response and then T cell response. And this is an important and vital information for vaccine producers. So what it means is that when you are producing vaccine for Lassa, you should start thinking of booster dose, because it means that the immunity over time. So because what we see is that this natural immune response from these survivors doesn't exist after 10 years. So when you design your vaccine, you start thinking, is there a need to put a booster dose after 10 years, since those that make it natural do not have it after 10 years? I think those are some of the findings and take home. And then I think the last thing I would say is that this study that was funded by this BBSRC have now helped us to build sort of an immunological platform to conduct some of these immunological assays. So when COVID happened, it was easy to transfer some of these tools into studying COVID. And we now have immune assays to look at also the type of immune response which Nigerians made during the COVID outbreak. And that we are working towards publication soon. Thank you.
Thank you so much, Dr. Ugwu. I think that's a whole lot of findings and information. So well done to you and the team. I think the first thing I want to ask is that observation, the unique observation that people who come down with the disease and people who are just exposed to the virus. Maybe I think you mentioned that for someone to come down with the disease, it's possible they were exposed to maybe a higher level of the virus that was capable of making them sick, showing symptoms, and then for the other person, it's possible the amount of the virus they were exposed to was not maybe high enough or significant enough to cause this. So I just wanted to find out, first of all, if this finding has been observed in other diseases, like you just talked about, transferring some of the skills, knowledge, and the techniques you developed for this research to studying COVID. So were there similar observations with COVID, for example, or other disease that you probably worked with?
Oh, thank you. Yeah, there are. In fact, some of the things we also found out from the COVID outbreak, which we are working towards publication soon, is there were people who did not everybody came sick with COVID, if you know.
Yeah.
Some people they had COVID and they were moving around, while others had a very serious, even some people died. So what we do know, or what we think, is that the level of virus you get also determines the kind of symptom or the response you will have. Now, do we know, if you want to put it in terms of number for people to understand, we don't know whether, let's say, if you have 100 virus, then you'll be sick. If you have 20, you will not be sick. We don't know. But the amount of exposure tends to also contribute, determine whether you'll be sick or not. And then also the state of your immunity. So similar thing we saw in Lassa fever, we also seen it in COVID, where some people are exposed at the same time while others fall sick and the others don't get sick. Now, part of the things we want to do is, now we've seen this, how do we unravel? How do we understand it? And this is our effort with this immunological studies. So what are the features of the immunity that is making me not to be sick when we're exposed to the same type of viruses while you get sick? So these are some of the subsequent works we are doing, both for Lassa fever and for COVID.
Okay, great. I am just curious, is it possible that maybe some persons did not come down with this disease because of their unique genetic makeup? Maybe because of their genetics, their immune system was able to wrestle the virus and subdue a disease?
It is. I will say it is. And this is what I'm going to say. Okay. So there is a study by my PI before I joined the lab long ago, where they looked at the Lassa gene and some people in, I think, some part of Ibadan, I don't know the exact places. And they realized that there is, I think it's one particular gene called a large gene. This large gene is involved in immune response. I think that's the best way to put it. It's part of the pathways of the immune response. And what they saw is that these people don't really get Lassa fever. I think so. I don't remember where it is in Ibadan, but I think that paper was probably sometime in the early 2011 or 12, and they found this gene that they think is associated with Lassa fever protection.
Mm-hmm.
That studies, we still hope for one day we'll replicate, but this is what is called a genome-wide association study, where you sample a group of people genes and then make correlation. And what they correlated is that there's a natural selection for the large gene, and this also makes this group of people not to fall sick. So what the point I'm trying to make is definitely there will be a genetic side to this. There's also an immunological side to this. But these are some of the things now with this whole interest in Lassa fever disease that many of us and other groups are trying to understand, to see maybe those that don't get sick, maybe there's a genetic makeup. For example, they found out this large gene is what is making them not to fall sick, or maybe just because that's the way they are, or maybe they just have a good immune system by the time they were engaged, they got the virus. And mind you, it's on record that in an outbreak of Lassa, about 80% of the people are asymptomatic. They don't get sick. So about 20% are the ones that actually get sick. But the problem with that sickness is that sometimes it can be fatal and severe. That's a challenge. Now, why is 80% not getting sick? Why 20% get sick? We don't know. And these are some of the things that we are hoping that with all of this interest coming along, a lot of people will engage, and we can find out in the near future.
Yeah, thank you so much. So I think that's the beauty of research, that there are always questions to still ask and answer. So yeah, I think my next question would be, so what are you working on currently before we delve into other aspects of the episode?
So thank you, because I feel like last time dominated my conversation. So like I said, with the platform we developed in the Lassa project, when the COVID happened, it was easy to translate most of our work to understanding the immune response to COVID. And we are lucky to have a seed fund from Cambridge, Africa, to look at what are the features of immunity from survivors of COVID in the early days. And that helps us to provide preliminary data. And luckily, the Bill and Melinda Gates Foundation were happy with what we were doing, and they also decided to support us to see if we can then build an immunosurveillance platform that will complement the genomic sequencing we'll provide from our center. So the idea is we generate all the genome of these viruses, and then we tell you this is the strain, this is the one that is circulating. What next? Can we support this genomic data with functional immunological data? Can we tell you, so this strain can induce this type of antibody response, this strain can induce this type of T-cell response. So this is what we're doing now, is building this platform. And it basically involves not using the same basic immunological tools, ELISA, Luminex to measure the different kinds of antibody response, measure pseudotype assay to measure neutralizing antibody response. And also, the next phase is now when you see neutralizing antibody, can you isolate those antibodies? And that's what we also, so can we also generate monoclonals for therapeutics for some of these diseases? So much of the thing I'm doing now is building this platform. And we've made enormous progress. So in the sense of building this platform as well, we're also using the platform, we're also using COVID as a test run for the platform. So we are looking at what are the features of immunity during the COVID-19 outbreak. So what kind of immune response did Nigerians make to the Delta variant, to the Omicron variant, to the Alpha variant, which of the variants? And what kind of immune response did you see from those that were vaccinated versus those that were not vaccinated, and those that had the disease, and those that had vaccine and also had the disease, which we call hybrid immunity. So these are the kind of things we are doing now. So we are measuring both the antibody response and T-cell response, and then in the long run, still supported by the Gates Foundation, to work towards generating monoclonal antibodies from our sequence data to see that will serve as therapeutics for both for COVID and also for Lassa. That's basically what we're doing now. And then we're also starting to look at, then once we finish the trial with COVID, we're hoping to use the system now to also study most of the endemic viral pathogens now, mainly providing immunological data, because once we have all the immune assay running, we can provide it. So for example, having a biobank of sera. And I think one of the biggest challenges we had during the COVID-19 is when the outbreak happened, most people didn't have sera where they can basically generate make new ELISA data with antibodies or the rest. So we are building a biobank for some of these endemic pathogens, like now Lassa fever and then SARS-CoV-2. So should in case there is a new outbreak, you don't have to look away. We can easily provide a sera that you can generate neutralizing antibody from. And so building a biobank, developing all this immune assay, and then hoping to translate it to other endemic pathogens such as dengue, hepatitis, yellow fever, and other endemic viral pathogens as well. So that's basically what I'm doing now.
Yeah, interesting. I think the transfer of knowledge, skills, and techniques is one great contribution your research has made to to understanding all the other diseases that affect us in this part of the world. When you were talking about the idea of being exposed to an infection, and your immune system reacts to an extent, although it's quite different from how it would quite different from a response that someone who actually came down with the disease would have. You talked about how it could inform vaccination strategies for this disease. So I would just want you to just comment on that a bit. If we have all the information about, this set of persons, they've been exposed to the virus and they didn't come down with the disease, and this set of persons actually came down with the disease and maybe they recovered, and this set of persons have never been exposed to the disease. So how can it inform how people are vaccinated, whether with COVID or with any other disease? Because I know with COVID, the idea was just, if you've not had the disease, come and take vaccines. No, there were no studies to assess whether or not you've been exposed to the disease, whether or not you've made, you have any kind of antibodies, maybe binding antibodies, or any kind of, whether your T cells were reactive to the virus. So how can, the whole idea of being exposed, not being exposed, or coming down with disease, how can it inform vaccination strategies or the development of vaccines?
So what is understanding what's the impact of existing immunity to vaccination. And I think that's what I understand. What I would say it's important to know. So an interesting case in vaccine is dengue. So there's what is called vaccine reaction in dengue outbreak, where because you're vaccinated, giving a vaccine to another one will cause an untoward vaccine reaction, inflammatory disease that can kill you. So it's good to know what's the impact of vaccination or pre-existing immunity to vaccination. Does it boost it, or do you need to let it wane before you don't vaccinate? Those information are very important. So immune system as well. Again, remember, I tell you, the immune system is such a well-organized and regulated system.
Yeah.
Less is a problem. Excess is also a problem for the immune system. So you could, if you have excessive antibody, the immune system also reacts to reduce the amount. So it also tells its own self signal that, hey, we have too much and we need to reduce it. If you have low, you also need to increase that way. So you need to also consider how do you perform the system to get the best response. For example, what we're now learning we learn every day from the SARS-CoV-2 outbreak is that initially we didn't know the impact of pre-existing immunity on the vaccination. But over time, we do know that those that have hybrid immunity, they have a stronger immune response. It didn't hinder the effect of the vaccine. So it's more like a booster effect from those that were infected and were vaccinated. And that's because it's possible you couldn't have had that effect. But what we learned is that those that had infection, it's okay to vaccinate them because they have a booster response rather than an untoward response. So it's important to have all of this, because if you consider that vaccination is a biological agent that you are given, you need to be, and again, this is one thing for those of us in immunology and vaccine field, you got to be extremely careful before you got to err on the side of it. You need to be sure what you're doing. And this is why sometimes people complain that why does it take so long to produce a vaccine? Why it takes so long is to be able to answer all of these questions before you start giving anybody any jab. So it is important to have those information to understand. We call them immune correlates of protection. Immune correlates of natural protection. If you understand what natural protection is, then you can then envisage how your vaccine will work, you see? And I think that's what I would say about understanding pre-existing immunity and how it will impact your vaccine. I don't know if that answers your question.
Yes, it does. Thank you so much. I think that was really clear and straight to the point. You mentioned at some point the idea that with a disease that has not been given a name, and let me put it that way, does not have a face, like so someone can be coming down with a disease, but we don't know what the disease is, and so we can just treat malaria or we can treat typhoid, and it's common in this part of the world. People just come down with symptoms and then they've already self-diagnosed and then they've gone to take certain medications. I like to think that it's possible that there are several other diseases that have not been identified in this part of the world, not because they don't exist, but because maybe there's no funding or nobody's really aware of those kind of diseases, or people are not giving attention to those kind of diseases. So I just wanted to just comment on that, especially with regards to how the field of immunology can help with making such findings, because if we've not identified diseases, it will be impossible to develop strategies or to tackle them. It will be impossible to do that. But are there ways that the field of immunology can help to discover new diseases that were previously unknown or that were previously just obscure to the world of science or obscure to humans? So how can the field of immunology play a role in identifying those kind of diseases? Because some of the diseases like Lassa, the Lassa virus, and then I think I also heard of Marburg, although they were kind of discovered in Africa, but it was because they affected foreigners. That was why it was easy to really spot these viruses. So with the advances we've made in science, advances we've made in research into infectious diseases and immunology in Africa, how can the field help us identify or spot diseases that were previously unknown or that have been misdiagnosed in the past?
That's a very big question. I don't know if I have the shoe to answer it, but I'm going to try my best.
Okay.
Immunology will definitely help. And again, I think one of the big challenges we have in Africa is that many of our politicians and leaders haven't seen the business of science. They only see science as charity. And when you start seeing the business of science and the impact, then it will boost investment in science. I think that said, I have to be positive here. We've made enormous progress. Here at ACEGID, we have a project we call Sentinel. And the aim of Sentinel is basically active surveillance, It doesn't, so I think the problem we've had over time, and that's why people were now discovering, is that we play defensive with the pathogen. We are reactive. So it's only when there is an outbreak that we start working. But I think we shouldn't be reactive, we should be proactive. So we need to start looking for this thing. We need to be offensive. We need to look for them and then find the best way to coexist with this pathogen. And that's one of the big or major objectives of the Sentinel project, where you proactively look for pathogen I think about 70% of diseases you have will come from animals as well. So when you then look for some of these diseases before they become even a disease, then you could understand it. And thanks to the power of genomics, you could do this now. You don't have to do the cut postulates to disease discovery. You can basically take from the sample to disease discovery by looking at the sequences. You see, and then which is one of the big ones, the WHO called the disease X. So you can just, with the power of genomics, you can move from sample to disease detection and identification, discovery. So it's because technology has made it easier, and some of us here at ACEGID are working hard and making the best. So I don't think we are where we are, but at least the contributions Africa made during the COVID-19 shows that we are prepared and willing to contribute if given all the necessary tools. And I'm going to say this thing again before I get to how the immunology will contribute is the world is so global that if Africa doesn't succeed, the rest will not succeed. That's the unfortunate thing that people need to realize. The rest can move on. Because first of all, look at how the COVID moves around globally in a short time. There is no disease that can move. Regardless of how much you put travel ban, whatever they put there, this thing will move. You don't control movement of little microscopic organisms. Your ban doesn't stop them. So It's good that there's enough capacity and investment on the continent so that we have the wherewithal to tackle this before it becomes a global problem. So waiting for the world to come and help us, it doesn't help because by the time they come, it's already a global problem. And now going to immunology, how can immunology help in some of disease discovery or in pathogen discovery? Again, with technology, what we are looking at, which is one thing again we are grateful to the Gates Foundation for funding this immunosurveillance platform, is so what you could do now is no longer COVID was a novel virus, but look at in a short time, you have a lot of molecular diagnostic tools. You have a lot of immunological assays to detect this pathogen. If you don't have diagnostic tools, then how will you know whether there is a disease or not? So what immunology helps us to develop some of this immunological assays to understand and discover some of these diseases. So for example, basic immune assay ELISA. So why we look at some of this surveillance idea is providing this immune data to support it. So those immune data, what can you do with them? You can use them to develop diagnostic tools to some of these pathogens. So a very practical example. There's an outbreak of yellow fever in Nigeria. There's no diagnostic tools. And then what we do know the patient that had the yellow fever and recovered. Once you collect serum from that patient, from that serum alone, you could develop ELISA to measure the antibody. You can also develop ELISA to measure antigen. You can develop Luminex to measure the antigen of the virus. In a short time, you can do this. So you see the power of that serum. And all of these assays I'm mentioning now are immunology, can only be developed from the principle of immunology and a good understanding of immunology. So this is where I call translational immunology. So using the immunological tools in providing solutions. I told you that one of the goals of ACEGID now with our immunosurveillance and GISA project is towards developing monoclonals for both SARS-CoV-2 and for Lassa. This is what is being funded by the Gates Foundation, and we hope that we can get there. There's no licensed therapeutic drugs for Lassa fever. I'm going to put it on for now. So most people use ribavirin to treat and other ones, and I think favipiravir, but there's no licensed drug now. So imagine if we can, from all of these are cohorts of survivors, and we're able to develop a monoclonal. That would be a game changer, especially for healthcare personnel. So why will I be scared again if I know that there's a therapeutic monoclonal I could take if I'm somehow exposed to the pathogen? So these are some of the ways you can translate immunology into solutions, and we are working hard to be part of those solutions, and I believe with more support, and I think that's the last thing I'm going to say is that we need to start seeing science as a solution business, and rather than a charity. And when we start seeing it that way, it turns out it will benefit everybody. It benefits your citizens. It generates more money to the economy, and everybody benefits. And I think that's what I would say.
Thanks for answering my question, and also thank you for bringing in the conversation of the need to see science as business that can bring in investment and bring solutions that can also come with profits. Yeah, thank you so much. I think I read one of your interviews sometime last year that you also highlighted that aspect of science, that it's important for us to just think beyond charity when we are thinking about research, but we should also look at how we can take proactive steps to explore the business side of science and look at how we can make money from it. Thank you so much for that, and thank you so much for your time. You've said so much already, and I think your research is really fascinating. And so we'll just take maybe final words from you now as we wrap up. A word of advice or two to emerging scientists, things that have helped you in your research and in your journey that can also be of help to other young and emerging scientists in the field of immunology.
Well, I would say I'm still learning, but I think this is my mantra. I normally say this thing: when you are being used, be useful to yourself, to the person using you, and to your community. I'm going to say it again. When you are being used, be useful to yourself, the person using you, and to your community. And I'm going to dissect it. Rather than complain of someone taking advantage of you, how do you turn out that time the person is taking advantage of you into making yourself, benefiting yourself, benefit that person? Because you can't stop the person if you can, and also benefiting others. So it becomes a win-win, and we will not leave you with regrets. The second thing I'm going to tell people is it's not easy doing science in Africa, at least from my experience in Nigeria.
Yeah.
But be patient, persevere. Be patient, persevere, and put in extra work. If you think it's not working, just persevere, keep trying, don't give up. It may look bleak. In 2 years, in less than 10 minutes, you become a global phenomenon.
Yeah.
So be patient, persevere, don't give up. Just keep going. Even if you don't know, just keep going. Believe in yourself and keep going. And put in the work. Hard work first before ego. Some people put ego first before hard work, and sometimes it doesn't work. Because if you put ego, and then when they call you, you can't defend that ego. Put in the work, and then ego will come. And then I think finally, the last thing I'm going to say is it's tough, because I also don't think I'm doing well with it, but find a way to maintain your work-life balance. At the end of the day, if the work goes, you got to find someone that you can look at and say, hi, how are you? So find a way to maintain your mental health and work-life balance against all odds. No matter how taxing your job is, have a very good balance. Make good friends, have good colleagues, mingle with people. If you want to have a family, just maintain a good life balance because there's life after work.
Yeah.
And it will be sad if at the end of the war you alone and there's nothing else. Try to have a good mentor is very important. When you choose your PhD, your supervisor becomes your father. Some people tell you to separate it. I don't think it works. Have a good relationship. If you can't have a good relationship with your PhD supervisor, it becomes difficult. I'm not saying you may not succeed, you will succeed, but mentor, pray that you have a good mentor, someone who genuinely care about your interests. Yeah, someone who genuinely care about your interest. Mentorship is important.
Yeah.
I had a good platform when I came back to Nigeria with ACEGID, but I'm so happy. If that platform wasn't that, I may still succeed, but it may probably take me longer. I had a great supervisor who took out, he never gave up for me, and he believed that I will succeed. And I think that's what is keeping me here now.
Many thanks once again to Dr. Chinedu for sharing those tips and discussing his research with us. One thing that comes to mind when you think about Dr. Ugwu's research is that we need to be looking in the right places if we are to understand and develop effective therapies for diseases. To understand Lassa fever, he had to study the immune responses of those who had survived the disease and those who had been exposed to the disease but did not become sick. And it's interesting that the same model can be applied to several other diseases that affect us in this part of the world.
Thank you for listening to this episode on Immuno Africa. If you enjoyed it, please feel free to share it with your network. You will learn first about future episodes and get other immunology related updates by following Immuno_Africa or the Immunology in Africa podcast on social media. See you on the next episode. Bye.